Hello, everyone. Thank you for coming. My name is Reynaldo, and today we will be talking about some Pseudomonas species, bacteria helping in the colonization and biodegradation of environmental microplastics. So, plastics have posed an increasing and concerning issue for the humans. Uh, since some studies have shown that we ingest around 5 grams of plastics every week. That's a credit card worth of plastic. Also, some many studies have found microplastics, which has small pieces of plastics less than 5 millimeters in diameter, and they are everywhere in your life. They are in soil, water, in the clothes that we wear when we wash it, in the tap water being 94.4% according to some studies. So this is actually a top priority issue that needs to be addressed. We are going to be using for this study microbes, in this case Pseudomonas statuary, which is a gram-negative biofilm forming bacteria. Uh, and we are going to go incubate it with some of three widely used plastic types: PVC, polyvinyl chloride, low density polyethylene, and polypropylene. And we have hypothesized that this co-incubation, in this process, the Pseudomonas statuary will colonize and biodegrade these types of plastics. So after we created the microplastics and co-incubated from 7 days to 180 days in time intervals, we have extracted at each of these collection points and run some tests to see if there is some colonization and biodegradation of the plastics. First, we transfer these plastics to the PIA, Pseudomonas isolation agar, to see if there is any colonization and growth. We see on the upper row no growth on the control samples as we expected, but in these samples at the 125 days, we can see clearly some growth of the bacteria around the plastics. So this is really good information showing that there have been some growth on the microplastic. But what is happening at the surface of the plastics at a microscopic level? And for that, we use the SEM analysis with a magnification of 8000x. We have the control on the top row again. We have our samples co-incubated with the Pseudomonas at 7 points, 7 days collection point. We have the biofilm is clearly visible here in these samples respect to the control, and is made by the bacteria. We also see these shapes, these rod shapes, uh, which are from the bacteria. These Pseudomonas are bacilli. So there is evidence of this colonization at the microscopic level. Now we wanted to know if there was any chemical changes uh, in the composition of all these plastics. For that, we use the FTIR infrared spectroscopy. Again, we have the control on the top, and we have these samples, co-incubated samples at the 56 day, 90 days, and 125 days for plastic 3. So we can see some differences on the stretch and the peaks. First, in the 3300 and this area over here, we can see some peak. This could be attributed to the OH alcohol stretch, and then just before hitting 3000 we can have also some changes in the peaks. This could be corresponding to the NH stretch. So we know that this kind of bacteria is a denitrifier, interacting really well with the nitrogen in the environment. uh, Another example we have over here, 1600, some difference in the stretch. This could be the CO carbon oxygen double bond. Again, we have many multiple changes here in this stretch and peaks. So there is definitely something happening. We have again some positive and similar results that indicate that there is evidence of colonization, biodegradation of the microplastics by these Pseudomonas statutory bacteria. So for future studies, we are planning to use the SEM FTIR to analyze the last samples, 150 and 180 days that we couldn't because of the COVID-19 pandemic last year. We are planning to include some other bacteria such as fluorescence and alkaligenes, and we will use nutrient deprivation, exciting process. We are hypothesizing probably that with giving less food to the bacteria, we might obligate it to use the plastic and to eat more plastic, using it as the source of food. So thank you very much for sticking. Have a great day.